Marchantia sporophyte. Marchantia thallus. Marchantia archegoniophore. So here you can get the archegonia like this elongated one, neck region, so that you can identify it is an archegoniophore, longitudinal section. Here you can observe the antheridial structures. So this is the Marchantia antheridiophore, longitudinal section. So here this is the cup-like structure. So this is the Gemma cup of Marchantia. This is the Anthoceros sporophyte, longitudinal section. Anthoceros thallus. Anthoceros antheridia, thallus with antheridia. This is Polytrichum structure. This is the opercular region and peristomial region, and lower region you can get the capsule, but it is the low power diagram you can observe like this and under dissection microscope. So this is the dissection microscopic structure. Polytrichum sporophyte, longitudinal section. This is Polytrichum antheridial head. Polytrichum archegonial head, the upright diagram. So this is the venter region, neck portion. You can easily find out the archegonia. This is, see, please observe the sporangia. 3 are there in this diagram. So the Xylotum synangium, this is the slide. Selaginella stem, because 2 of the steels you can observe, even 3 of the steels in a line arranged, so that you can identify it as a Selaginella. This is the Selaginella cone, megasporangia, microsporangia, both observed in a single region. So T-shaped peltate heads you can observe here, that is the Equisetum cone longitudinal section. This is Equisetum transfer section, cones transfer section. Even here also you can observe the T-shaped structures. This is the peltate head, and lower region you can observe the sporangia. This is the Equisetum stem TS. This is Teres sori structure. Here you can observe the sporangial structures and the broken open one. With spores you can observe sporangia, and this is the pseudoindusium as well as the Teres leaf structure. And this is Marsilea HLS. You can observe the ventral upper side vascular bundle, and here in 2 rows the indusia you can observe, the receptacle as well as megasporangium, microsporangium you can observe here in this structure. Even the wall of this sporocarp is very, very thick. You can easily find out this is as a sporocarp of Marsilea. This is, please observe the aerenchyma at the peripheral region. So this aerenchyma representing the Marsilea structure, and this is the xylem. Both sides phloem you can observe, that is why it is a cylindrical or the amphiploic siphonostele structure, which you can found in the rhizome of the Marsilea transfer section. So this is the algal zone. You can observe the nostoc or anabina here in this region. Even you can found the tannin structures as well as gelatinous walls cells in this region. So this is Cycas coralloid root transfer section. So this is also the same one. Cycas leaflet transfer section. Two of the slides we can have, and this is also another coralloid root structure. Both two slides available in the lab. This is also a leaflet of Pteris. The ovule of Cycas, sorry, not Pteris. Okay, please, we will go through with the. So this is leaflet of Cycas. Even you can observe the horn-like stomatal region where the subsidiary cells which are looking like horn-like, and this is the ovule of Cycas. And please remember, this is in the small microscope or the dissection microscope structure. This is the Pinus needle transfer section, and this is Pinus male cone structure under the light microscope, and this is under dissection microscope structure. Two of the slides we have, both are looking like this, and this is the female structure. Even though you cannot observe the ovuliferous scales, but the ovule structures you can found here in this diagram. So this is the dissection microscopic structure of female cone of Pinus. This is the female cone of Neum. So axis, the cupule and the ovule structures you can observe here. That also in under the dissection microscope. This is the male cone of Neum under microscope, normal microscope or the light microscope. So this is the cupule, and here you can observe more than three layers of male flowers in the longitudinal section. This is also under light microscope, the Neum male cone longitudinal section. This is obviously the human skull-like which is present in the leaf portion. So it is a monocot leaf transfer section. And this is because many exarch radial vascular bundle you can found. Exarch means root. So this is the monocot root transfer section. This is monocot, even though you can observe the human skull-like. So obviously this is the grass leaf. Even you can observe the motor cells here and there. This is the upper epidermis and this is the lower epidermis. So this is the grass leaf, and a peculiar young stem, dicot stem transfer section. You can also observe the trichomes in the, this is, see, please observe the upper palisade parenchyma, lower spongy parenchyma and C-shaped vascular bundles. So this is the dicot leaf transfer section. So two of the slides we have, this is also another one, and here also you can observe the spongy as well as palisade parenchyma, the C-shape like vascular bundles. So if such a type of structure is there, you should be identify it as the dicot leaf. And it is, so please observe here, larger vascular bundles arranged in a single row, and the smaller ones are arranged towards the periphery, and sclerenchymatous ring is present towards peripheral region, so that you can identify it as Boerhavia stem. This is also Boerhavia stem. Two of the slides we have permanent slides in the lab. So this you can observe the Dracaena stem. Please observe here and there the rhaphites you can observe here, smaller round structures that are filled, as well as the needle-like structures, rhaphites that are present usually, so that you can identify it as Dracaena stem. And this is, please observe the xylem with the spiral thickenings, even pitted thickenings and reticulate thickenings you can observe here. So this is the xylem varieties, the thickenings of lignification or thickenings in the longitudinal section of the xylem. Thank you.